Today I have the honor to present Yoram Ben-Shaul from the Data Medical School. Yoram did his undergrad studies in Hebrew University, studied in Biology. Later on he did his PhD studies in Moshe Aveles lab. Later went to Harvard University to do his postdoc in Katrin Zulak, where he developed, probably going to mention it in your lecture, he developed a way in which to record from the ancestral faculty above and activate something that needs to be activated in order to record. I will talk about more details later. We used this development to open here a lab in 2011 in the Data Medical School, and that's it, I think. Let's hear what has to say.
Thank you. Thank you, Amit. I should say that before being at Harvard, I was also at Duke with Larry Katz, who passed away, and then I moved on to Harvard. So thank you for coming. When I was about to prepare my presentation, I wanted to look what was the title that I gave to Amit, and I realized that I said that I will talk about encoding socially relevant information by the mouse brain system, and I didn't understand what this means. So if you came here to learn about the brain system, I have to disappoint you. I'm not going to talk about the brain system. Just my brain system wasn't working when I gave Amit the title. So I'm going to talk, you can choose your title, either information by the mouse brain or by the mouse glomerulonasal system. So I will tell you about the basics, things that we started doing in the lab, things that some of you may have heard about, and then a little beyond, things that we're doing now and are more work in progress, and they're going to be preliminary, some of them. So I'd like to set the framework for what we're studying, what we're interested in, with this general scheme. And the one sentence that the catchphrase for what we want to do in the lab is to understand the computations that underlie social behavior. And we do this in the context of chemosensation, smell, olfaction. So this is an important fact. And here's the general scheme. So ultimately, what we want to know is we want to get information about certain animals in order to direct appropriate behaviors towards them. So we want to get information about some characteristics or traits that these animals have. For example, here are some red animals, some red mice, and we want to know that they're red because we may behave differently to red mice than we would to non-red mice. So red could be the sex or the age or any kind of trait that you may think of. And because this dictates behavior, we can assume that somewhere in the nervous system of the other animal, something in the neuronal activity can reflect this specific trait. So here in this very simple example, I just showed neuronal activity, uncharacterized abstract neuronal activity. Something is associated with this red trait. And to be detectable, for the nervous system to detect this red trait, there must be some unique chemical characteristic of this trait, of this feature. And here I plotted, again, a simple three-dimensional chemical space where you have three molecules, and this represents, each dot represents some concentration combination. And again, in this hypothetical example, what is unique to the red animals is some ratio between two of these compounds, C1 and C2. So this is just an example of what could be a chemical code. That is the way that information about a certain trait is encoded, if you will, in the chemical signature that is emitted by organisms with a certain trait. And of course, these chemical profiles are a result of some metabolic pathways or some genetic pathways that are characteristic of these specific animals. And the neuronal networks that have to decipher this information are somewhere in the brain, and they are the ones that are able to obtain these signatures, retrieve the information from the complex chemical cues, and translate them to neuronal activity. Now, to fully understand the whole process of chemical communication, we would need to understand, in my opinion, we would need to understand all of these aspects. And of course, we're very far away from this, and people are studying each of these aspects, but we're not studying all of them. What we are doing, basically, is running experiments where we measure the responses, neuronal activity in various regions of the brain, in response to stimuli from animals, and we can control the traits, that is, the features of the animals that we're presenting stimuli from. And eventually, we're trying one second to infer what these networks may be doing, but there's a big logical jump that we have to make when we try to interpret the activity to understand what the networks are. I'd be already a question.
Yes, I'm just trying to put it in perspective and in analogy to the visual system. So you're looking for things like, I don't know, orientation tuning in the olfactory system or something like that?
No, I'm looking more like face recognition in the olfactory system, I would say.
Higher in
The way that I pose the problem, it's based. I'm thinking of something that's already enough to direct behavior, and there are several stages in that in order to reach that information. Yeah, to reach this abstract information. And so, more specifically now, we studied chemosensory coding by focusing on the vomeronasal system, as Amit just mentioned. So here you see a diagram of a rodent head, and you see a scheme showing the main olfactory or the flow of information in the main olfactory system and the accessory olfactory system. I'm actually not planning to give a detailed introduction. I'll assume that you know about the main olfactory system, and I will say that the vomeronasal system, also known as the accessory olfactory system, is similar in the sense that there are sensory neurons in the nose. They send their axons to the olfactory bulb via the vomeronasal nerve that you see right here, and they reach the olfactory bulb, a certain region, specialized region, called the accessory olfactory bulb. And from there, information is transmitted further downstream to the amygdala. This is also something that's reminiscent of the main olfactory system.
So those are two systems?
Yeah, two different systems. Eventually, they converge. No, you don't have this system. Humans don't have this system, at least not functional, but most animals do have them. And mice certainly have it. Well, the sensory neurons are in the nose, yes. And the processing is in the brain, yes. I will talk about the organ in a second. And there are many ways we think about the differences between the two systems. I think that one general statement that we can make is that the vomeronasal system is specialized in processing stimuli from other animals. Okay, I'm not saying pheromonal communication, I'm saying from other animals, which is a more general definition. And as for your question, the sensory neurons, and that's another important thing to know about the system, the sensory neurons are located within a very unique organ called the Vino, the vomeronasal organ, which you see right here. You see one of the vomeronasal organs. There are two, one on each side. And this organ, this Vino, again, I will not talk about its function in physiology, even though it's fascinating. It's enough to know now that it functions like a pump that can suck liquids from the nasal cavity, and if these liquids happen to have ligands that can activate the sensory neurons, then this information is detected by the system and relayed to the brain. Okay? And the physiology of the VNO, it's a slow pumping process, is largely responsible for the slow dynamics that you see in the system, slow dynamics of responses, which I will show you in a few slides. So in our experiments, stimulus delivery involves not just it's not enough to apply a stimulus to the nose, but you also have to stimulate the VNO, and we do this electrically by stimulating a sympathetic nerve trunk. This is the system that I developed during my postdoctoral work. So in this talk, as I already said in the beginning, I will first talk to you about a story that many of you have heard already. I will maybe have some new insights and some new analysis, but it's essentially the same story, and that is about extracting chemosensory information about particular traits. And the little beyond will be something about the temporal aspect of the responses in the system, and also about plasticity in the vomeronasal system, which is something that I think is very exciting that we're starting to get data about. So this is a scheme of our experimental preparation. The VNO is here. This is a sagittal section showing you the AOB, and this little red thing here, or the red thing here, is a tract of one of the electrodes. So we're recording activity in the AOB, the accessory olfactory bulb, which is the first brain relay of the vomeronasal system. This is the region that receives the axons of the sensory neurons in the VNO, and we're activating the nerve to get to, as I said, after we put some stimuli. And this is a picture, this is a drawing, which shows you much more realistically what the AOB looks like. And in the AOB, there are several cell types, of course. There are glomeruli, like the main olfactory system, but here they're relatively disorganized. And these cells that you see here are the mitral tufted cells. They collect information from the sensory nerves, and they transmit this information further downstream. And in our recordings, we try to target them, because they are the ones who are very important in the circuit. They are the ones who transmit the results of the computations downstream. So our electrodes are targeted to these to this region. And here's a recording. This is a 15-second recording showing the low pass filtered signal. So you can think of our electrode. This is our scheme of our electrode array. It's 8 by 4. So we have 32 recording sites. They form a plane, and this that shows you the spatial relationship between these sites, not to scale, but this is a spatial relationship. And what you see here, these are 15 seconds. Of course, this is simultaneous recordings from all the different sites. This big thing right here is the stimulus artifact due to the stimulation of the nerve that I mentioned, this whole block here. And you can see that after some time, something happens here in the system. There's some increase in the local field potential energy, and it's not restricted. It's not in all of the sites because we're not recording from the AOB from all of the sites, but only some of them. And this is actually something that we only started looking at now, and I actually do not know yet what these frequencies exactly represent. But what we do is we look also at the spikes, and these are the spikes plotted on top of the LFPs. And you can see, for example, that there is an increase in the occurrence of spikes after some delay following the nerve stimulation. I should add that before stimulating the nerve, we also applied some stimulus. In this case, Anat applied predator stimulus to the nostril of the mouse. So this is the response to the suction of the stimulus into the VNO. Yes? We believe that these are microcells. Yeah, we think that these are microcells. We're not 100% that all of them are, but we verified that the electrode tract is within the layer that contains them. Colors are just different spikes. These are sorted spikes, so each color is one cluster. Yeah. This is an anesthetized mouse. Yes.
And that's the reason you need the stimulation.
Right. So okay. Since you asked, yes, the VNO is not unlike breathing, which happens spontaneously all the time. The vomeronasal organ is not active all the time. It's not. It's active during times of arousal, most likely, and when the mouse is anesthetized, it's not so aroused, and so we have to induce it artificially. And this is the thing that we do. We stimulate the sympathetic nerve trunk, which happens to innervate this organ, and this is why we have to do it.
Kind of like a smooth muscle that pulls the liquid in, or
Okay, but Amit would have to add this time to my talk. So this is the VNO, and this is a coronal section to the VNO, and it has two compartments. So this is all within, this is the left VNO, the right VNO, or the other way around. And this is all in a rigid capsule, okay? And the VNO is a blind-ended tube. It's open on one end to the nasal cavity, it's closed on the other, and there is a lumen. This is the lumen, which contains the sensory nerve endings. And in addition to the lumen, you have this blood vessel. And since they're all encased in a rigid capsule, when the blood vessel dilates, this shuts down the lumen. And when the blood vessel constricts, it opens the lumen, and then it causes suction from the nasal cavity. And this is what the sympathetic trunk activation does. It innervates the blood vessel and causes this constriction. So this is how it works. I skipped over this. I apologize.
So like a summer allergy.
It's what?
Like a summer allergy.
Like a spring allergy, yes.
Right.
Spring allergy, yes. Okay, so these are recordings, very old, well, not very old, but very early recordings made from the accessory olfactory bulb. And here you can see that AOB neurons can respond to and can distinguish between different species. So these are responses to a bobcat and to a fox and to a rat. And each row shows you one neuron, and you can see that it can have a selective response to one of these species. And you could even go at higher resolution, of course. And these examples, you can ignore the rasters now and look only at the PSTHs right here, shows you two neurons, one of which responded to female mouse urine, the green one, and one which responded to male mouse urine. And you can see that these responses are selective. So this is a very basic slide showing you that accessory olfactory bulb neurons can respond selectively to different species and also to different sexes within a species. Now, our present goal is more challenging than that. We want to look at something that's finer, and we're going to look at detection of two different traits. And the traits, or the characteristics that we will look at, are the identity of another mouse and the reproductive state. And we're going to look to do this in females. So the reproductive state, by that we mean the estrous state, whether a female mouse is receptive or not to reproduction with a male mouse. And Both are important to direct behavior, and I think of these as two orthogonal properties, traits, because for any individual, female can be in one of the two states, right? And so having one trait does not give you any information about the other trait necessarily. And I'd like to spend some time with this maybe confusing slide and to explain why this is not a trivial problem, why trait detection is not something that should necessarily be so simple. And even if we think about a very, very simple code, and the simplest code that I can think about is that you have one molecule whose levels will indicate whether the female is an estrus or not. So here's this scenario that I show here. We have the red trait. You can think of this as an estrus, and you can think of the black trait as non-estrus. And this, the horizontal axis, represents the concentration of some compound, hypothetical. This is all hypothetical now. And this is the probability distribution. So in this example, it's very easy to separate. You can, by looking at the level of the compound, you can set some threshold, and of course it's easy to come up with a neuron that detects that, and to separate and identify whether this was a red or a black mouse in terms of this trait.
This would be a single neuron that will respond to...
In this case, yes. In this, I'm just saying that it could be a single neuron, but I'm not saying too much about the network now.
Neurons don't behave like that. No? Normally you have some sigmoid that responds to...
No, these are not neurons. What are you talking about, the green or the black or the red?
So you're representing a population of neurons or a single neuron?
With what? With what? The red?
No, both.
No, the red and the black are not the neurons. The black and the red are they're the distributions of the concentration of this compound C. There's one probability distribution for the black mice. It's a Gaussian in this example, and there's another distribution shifted to the right, so it's a distinct distribution. It's like a distribution of heights of people. Of kids have a certain distribution of height, and then grown-ups have another distribution, and by studying its record, you can distinguish them. This is it.
Okay. I would expect it to be continuous.
What?
Based on the...
Well, I'm giving you a hypothetical example that it's different for the two different traits. With height, it would be continuous. But this is just an example that the goal is to explain why this is... Yeah. What?
Different molecules, or it's the same molecule?
No, it's one molecule at the different concentrations. If it's... Okay. Now I want to show you the same thing, but looking at now, to confuse it even more, now we're going to look at the concentration on the horizontal axis, okay? And again, these are the same distributions shown right here. And so this is the green threshold that we had before. I didn't want to reveal this yet. And now what happens if we play with the stimulus dilution, as often happens in nature, right? The stimulus can be more diluted or can be more concentrated, for example, due to evaporation. So if the stimulus is more diluted, then all the concentrations will be lower, and the threshold that we had for the original secretion or stimulus will not be good anymore. Or we can do the opposite, and we can look at a more concentrated solution, and again the same thing, except that everything is shifted upward, and our threshold will not be sufficient. So all I'm saying is that one simple threshold cannot always provide information or reliable information about the trait if it's encoded by just one compound. And here is another example. Suppose we're now looking at another strain of mouse, okay? And here again, the red is maybe more, the red trait is associated with higher levels, but everything is shifted. So again, I would need a different threshold to distinguish these two. And in some cases, you may even think that in different strains, or maybe in different secretions, because remember, an animal can have different sources of secretions. There's saliva, there's urine, for example, in the case of mice, and maybe the distributions are different in the different secretions. Making all of this more complicated and showing that one simple threshold cannot be used to discriminate this specific trait. And all of these examples, they may seem very hypothetical, but there are chemical analyses that show just that. That is, that chemical distribution of estrus and non-estrus cues are changed in mice of different strains, and sometimes even change the direction of change in one strain compared to another strain. So I hope this is clear. I hope I convinced you at least that it's not such a trivial problem, and we cannot expect just one sensory neuron at least, or one neuron that receives information from the sensory neurons, to be able to tell us under various conditions whether an animal has a red trait or a black trait. And this is, again, I remind you, this is a simple scenario of just one molecule. So this was the method that I just said, no need in repeating it. And Anat Khan, who is sitting here in the audience, a postdoc in the lab, set out to address this question. And the way she set out to address this question was by measuring responses to stimuli from 4 different types of animals. So we have 4 female mice here, showing the source of the stimuli. And you can see that 2 of them are black, they're the C57 strain. 2 of them are gray or white, they're the Bob C strain. And for each strain, we have an estrus and a non-estrus female. So we have a total of 4 different basic stimuli. And we're going to look at the reproductive state. We're going to look at the strain. Another thing that Anat was looking at is different body secretions, which I just mentioned. Not only urine, which is a very commonly studied source, but also vaginal secretions and saliva, which may also provide information about these traits. And in addition, we're going to look at these stimuli at different concentrations, just like in the example that I gave you before. And operationally, what this means that you cannot test all of these different sources of variability in one data set, so it had to be divided. So Anat had 3 data sets. For example, one of them was dedicated to vaginal secretions for each of the 4 stimuli, each of them at 3 different dilutions, low, medium, and high. These are the concentrations, low, medium, and high. Another set with urine, and one set also had a combination of the 3 different secretions from each mouse. I will mainly talk about this data set today. I will not talk about all of them. So first of all, these are some example responses, 12 different neurons that you see here. And what this example shows you is that all the stimuli that we tested, that are not tested, namely vaginal secretions, urine, and saliva, they can provide information about both the reproductive state and also about the strain of the mouse from which the stimuli came. So these are, the panels show you all the different combinations, but for example, this is one neuron that responded to vaginal secretions, and it increased its firing rate in response to vaginal secretions from a black, that is from a C57 female, but actually suppressed firing a little to vaginal secretions from a Bob C female. And another example is from another neuron that responded to saliva from non-estrus mice, but did not respond to saliva from estrus female mice. Okay, so this is just to show you that information about these traits is contained in these secretions, and accessory olfactory bulb neurons are sensitive to this information. And now we want to address this problem more formally. And when I talk about perceptrons, and Chaim is in the audience, I'm a little bit nervous, but I will do my best. So we're trying to decipher the source to understand what was the trait that the stimuli came from using a classifier. So here's the classification scheme. So we're looking at responses of individual units, and we're looking at a trial-by-trial basis. So for example, here is one response shown by red, and we're going to do something very crude. We're going to characterize this response by one number. This is the change in firing rate in a pretty long window, 40-second window, following the stimulation. So what we have is, for each stimulus, we have a set of numbers corresponding to the response of each of the neurons that we recorded. In different data sets, we had different numbers of neurons. And then we're going to use these numbers to try to guess what the stimulus was. Okay, and this is just showing you 2 different classifiers. One may be used for the state, one may be used to apply to understand what the strain was. And the idea here is that they may have different weights. That is, different neurons may have different contributions to different types of discriminations. I will show this with a little bit more detail. So what we're trying to play here, we're trying to pretend that we're a downstream neuron that reads information from A or B mitral cells. This is the classifier's decision rule. This is the Perceptron decision rule. And just for those of you who are not familiar, so the Perceptron is a simple linear classifier. It's a very basic one. It's not even supported in MATLAB anymore because it's so old-fashioned, but it's still useful, I think. And first, you train it for a specific classification using a certain part of the data set. And then the process of training finds the best weights, as I said before, to make this particular classification. And then we test the classifier on another subset of the data, just to tell you that there's a training set and a test set. And today I will focus on reproductive state detection. Okay, I will not talk much about strain detection. So let me start showing you the results of the classification. So we start with the simplest comparisons that we can think of. So here are the 12 different stimuli, the 4 different animals that I mentioned before, and 3 different dilutions for each animal. And the simplest comparison that I can think of is we look at vaginal secretions. We know that these are vaginal secretions at a given dilution for a given strain. So for example, comparing this one to this one, they're both at high concentration. They're both from the black strain, the C57 strain, and they're both from vaginal secretions. So the only thing that differs between these two is the reproductive state of the animal from which the stimulus came. We're going to try to guess the reproductive state, and we're going to do this procedure that we're going to start out with all the neurons. We're going to feed all of them, the responses of all of them to the classifier. And then, once we're done, we're going to remove one unit. We remove the one unit that has the least influence, and we keep on doing this procedure until we remain with only one unit. And you have 6 such comparisons here, but I'm showing you the average of all of these. And when you look at the performance of the classifier as a function of the number of units, it's almost perfect until the very last unit, but generally you can say that you can get very good classification even with a single neuron for all of these pairwise classifications. So this is an easy task, and it's maybe not surprisingly easily accomplished by the neurons that we recorded from. Now, one of the questions that we can ask is, once we have trained one classifier, does it work well on another dilution? I don't know if you can see these arrows right here, but we can take this classifier, this is not a classifier, this is just a comparison, but we can take the classifier that was trained to make this comparison and ask whether it does just as well on another comparison in which the only thing that differs is the dilution. And when we do this, you're going to see again the average performance across all comparisons, and it's very poor. It's almost at 50%, which is chance performance. So there's not a lot of similarity in the rules that one of these classifiers, or a lot of similarity between the rules learned by these different classifiers. So the question is, can we train a classifier to perform well across all of these dilutions? And that was the next thing that we tried to do. So here we have 2 different classifiers. So for example, this one right here has to classify estrus from non-estrus, but now we want it to do it regardless of the specific concentration of the stimulus. Okay, so we're going to present one of these 3 or one of these 3, and the classifier will have to decide whether it was estrus or non-estrus. It's a more challenging task now, right, because it has to generalize across the dilutions. And this is again the performance of the classifier as a function of the number of units. It starts out, the maximum that we can achieve is a bit more than 90%. And as we decrease the number of neurons, the performance falls off, and here the fall-off is more dramatic than it was before. Before, we maintained pretty good classification even with 1 unit. Here it's more dramatic. So best performance is lower than 100%, and multiple units are required to achieve it. And now we can ask the next question. We can ask, okay, so we trained one of these classifiers across all dilutions for C57 mice. What happens now if we try to generalize it to the other strain, to the bulb C mice, okay? And we can do this both ways. And this is the result of this generalization attempt. Again, average for the 2 types of generalizations, again, it's very, very poor. Meaning that training on one strain is not enough to make a good decision based on another strain. So this is a bit tiring, but this is now the last stage of this analysis. Can we train a classifier to detect the reproductive state across both dilutions and across strains? So here, now we're trying to compare all of these to all of these. That is, we'll give the classifier one of these stimuli, and it could be one of 2 strains, and it could be one of these 3 dilutions, and it will have to tell us whether it's an estrus or a non-estrus female. And this is the performance of this classifier. Again, you see the same trend that performance falls off, but compared to the other classifiers, the best performance still falls more dramatically with the number of units, and even more units are required to achieve the maximum performance, which is not as high as before. So just to summarize all of this together, now I'm looking at the best performance that we can achieve in each of these different classifications. So this was a simple pairwise. This is across all dilutions within a certain strain, and this is the very general one. And you can see that there is a fall in the best performance that you can achieve, and not shown here, but I showed you before, one second, that the number of neurons required to reach this best performance is larger with the more difficult classifications. Ken?
So you could give a mouse, these are female or male mouse you are studying?
These are male mice recorded from.
So if you give a male mouse the two odors or liquid or whatever you're using, how well can they perform? Like, you train them to tell you whether this is estrus or non-estrus female mouse. How well do they do?
Well, it's a good question. Well, mice can distinguish. Okay, mice can distinguish estrus and non-estrus urine. This is established, well established.
They can, when there is a female mice, like they can tell what state
Right.
this mice is, but it doesn't mean that they can only rely in this type of secretion, in one type of secretion. Right? If you are using urine.
I agree that I don't know that the information is only in one type of secretion. Well, but okay. Well, mice can also discriminate based just on urine. This is another experiment that people have done.
They can do that. How well? 9%?
I don't know, and I agree that a link that is missing here is a direct link with behavior. I also don't know how well mice can generalize across all of these things. So I'm now assuming that mice can do it, and it's true. It's true. It may well be that mice, for example, can better discriminate the estrus state in black mice as compared to the white mice. Okay, so there's a link here, and we're basing all of this analysis on the known, but it may be more subtle than we know, on known fact that mice can discriminate, and we also know that mice can discriminate the strains of other mice. So these are two things that are the basis for our work, and there's some assumptions here.
Because you're saying, well, I mean, they can do better than 90% using.
I'm sure mice can do better than 90%. I'm saying that given, and I don't want to make a specific point about the exact number. I'm just saying that the more general classifications are more difficult. They require more units. Yeah. I don't know. Okay, and yeah, the answer is I don't know. And so now let me try to make some sense of what I've shown you. Maybe it's obvious to some of you, maybe it will not be obvious even after I explain it, but I want to try to see why. Yeah.
Regarding the data, how sparse is it? So what's for each neuron out of these 32, you had an array of 32 electrodes.
Yeah.
What's the probability of each neuron participating in a particular stimulus?
So we have a distribution of the number of stimuli that each neuron is responsive to, and most of the neurons are significantly responsive to one of the stimuli. Okay, we have 12 different stimuli, and it falls off pretty much like I don't want to say exponential, but falls off like this. And then of those 92 that have a response, I don't remember the numbers. I don't know if Anat remembers, but I think it's about threefold. They're about 1/3 of all the 1/2 to 1/3 of all the neurons that we recorded from this region. So it's not like all of the neurons respond, and most do not respond to all of the different stimuli.
So in the more general case, how many states you have,
How many states?
Yeah.
Two states, either estrus or non-estrus.
How many different patterns of firing?
I'm not sure I understand.
How many different firing profiles you have?
If I look across the different,
How many stimuli you have?
12. So each,
Together, and you have, and
yeah,
look back at the.
each circuit is one stimulus. Sorry.
So look back at the curve of the performance. So 30 neurons are not able to distinguish, classify 12 stimuli.
Right. They're not able to take each of these 12 stimuli and say whether it was estrus or not estrus.
But the input to the
The inputs to the classifier are just not the entire PSTHs, but just the average firing rate.
Average firing?
Yes, which is shown here.
Average firing?
Yes.
There's no neural noise involved.
There is neural noise involved.
For each stimulus, you have just one vector finder.
Yes, but we have different trial repetitions, and they may vary a little bit from each other.
So you put a single trial, or you put a trial average? That's my question.
No, it's a single trial. It's not a trial average. Yeah, the average is over time. So we average over the 40.
It's a single trial.
Yes, it's a single trial, and there is variability.
So neural noise may be one limiting.
Oh yes, it is. And these are the nicer examples that I'm showing you here with the less variability. So there is neuronal noise. Okay. So now trying to understand why this is a complicated task. So what I'm showing you now are the responses of this data set for the vaginal secretions. We have 92 rows, which correspond to the 92 different neurons. And we have 12 different stimuli. So I'm sorry, it's a bit hard to see. 12 different stimuli. They're arranged into the estrus, non-estrus, and these are the bulb C, and these are the C57. Again, 3 estrus and 3 non-estrus. And the reason you have 3 for each is because we have 3 different dilutions, the low, medium, and the high. And this is, in this matrix, the responses are normalized. So for each unit, for each neuron, the lowest response is shown in green, the highest response is shown in red, and everything else is colored in intermediate colors according to the scale. And you should know that green doesn't mean suppression of a response, it just means that this is the lowest response that neuron showed to any of the 12 stimuli it was tested with. Okay? And this is pooling across the different experiments. And now what I'd like to do is to sort this matrix according to the weight that the perceptron, that the classifier assigned to each of these different neurons. So this is what you see here. These are not the exact weights, this is just a diagram. But now I'm looking at this classifier. This is the classifier that was supposed to distinguish estrus from non-estrus across all dilutions in the bulb C mice. And you can see here that the rows, it's important to say, at the bottom you have, at the bottom and at the top, you have those neurons that have high absolute weight. These are the ones that contribute to the classification. And you can see that the neurons that make a difference, that the classifier really listens to, are those that can distinguish between the estrus and non-estrus. You can see the red here versus the green here, and the green here versus the red here. So there's some sense in how they distinguish the two traits. Yeah.
How many mice do you put from?
This is about, I think, 6 mice in this data set.
So, and if you take each individual mice, you still have, so maybe all the, 5 best units are from one mouse, from the others. You check if one mouse can still hold the analysis.
Actually, one mouse can hold the analysis, because this was one reviewer's question whether one mouse can do it, and we tested it, and yes, one mouse can do it. It may be that this is the one mouse that holds all the, that is responsible for all the successful classifications, but as I will show you soon, this is not the case. Okay, we can remove many units and still do pretty well. Now I'm looking at the other classifier, and this classifier now does, again, reproductive state discrimination, but the difference here is for the black mice, for the C57 strain, corresponding to the rightward columns, and it's a different sorting of the matrix, and you can see here again that the distinction is between, you can see a distinction between the estrus and non-estrus columns on this side of the matrix. And the important point to make here is that, for example, looking at this reordering of the matrix, the partitioning to estrus and non-estrus distinction does not apply to the other strain. I hope you can see it. It works well here, but it does not apply to the other strain. And the same goes if you train specifically on the bulb, on the black C57 black. The partitioning to estrus and non-estrus responses is not apparent in the white mice. I hope this is clear, because this is a bit of a confusing point. And in order to achieve this, and this is why when you train one classifier and you try to apply it to stimuli from the other strain, it's not going to work so well. This is why you saw such a low success rate. In order to be able to generalize, as you saw in this classifier, we actually have to explicitly train with both strains. And then the classifier will choose those smaller number of units that can generalize. It's not perfect, it's noisy, but that can more accurately represent estrus state regardless of the strain of the mouse. So this is my intuition for why this is more difficult. So now what about the other comparison? So far I focused only on reproductive state across concentrations and cross concentrations or dilutions and strains using vaginal secretions. There are much more comparisons that can be made. We can ask, what about detection of strain? What about detection with urine? What about generalization across secretions? This is another thing that can be done. And you need not worry, I will not tell you about all of these right now. In all of these cases, the outcome is that trait detection becomes more difficult as more sources of variability are taken into account. So sources of variability are dilution, are the strain, are the source of the secretion. These are the sources that we tested here. I would like to point one example using vaginal secretions to detect strain. So this is a figure, this is an image that I showed you 2 slides ago. This is the ordering based on the generalized state detection. And now I'm going to look at this classifier that I did not talk about, but you can see maybe that its goal is, or it was trained to distinguish the white mice from the black mice. So it's a strain discrimination, and we are trying to ignore the ester state and the dilution from which the stimuli came. And now I'm going to show you the ordering of the matrix according to this classifier. And I don't know, maybe you can try to think what you will see, but here's what we see. Here's the weights, the units that get the highest weight. It's the same set of data in the 2 matrices, but just sorted differently. The rows are sorted differently. You can see that those units that had the higher weights that contributed to the classification are those that distinguish well between the 2 strains and not so much between the 2 reproductive states. And we can then ask, what is the relationship between the weights assigned by this classifier and that classifier for each of the units? And this is shown right here. This is the correlation between the weights. And the point is, for this classifier and also for other secretions and comparisons, that there is no correlation between the 2 weights. Okay? So a unit can have a high weight in one classifier. It doesn't necessarily mean that it will have a high or a low weight on another classifier. So a given unit can contribute to multiple classifications. So let me give you the conclusions thus far from this analysis. And for these conclusions, I'd like to use the old tale. I think it's an Indian tale originally, or maybe a Persian or Chinese. Wikipedia has various versions. This looks to me like it's in China, yes. And the story is of the men. These are blind men, and they try to understand what an elephant is. And each person feels a different part of the elephant, and of course, each of them has a different impression of what the elephant is. And the other way to look at it is, if you really want to make sure that these people encountered an elephant, you'd have to ask several of them what they felt. Just asking one of them may be ambiguous. And this is the analogy that I'd like to make. That the neurons, these are now, instead of the blind people, you have the neurons. This is a drawing I commissioned from my son, and each of the neurons has information about a certain aspect of the stimulus, and you really have to pull this information together to achieve a reliable discrimination or reliable identification of the stimulus, whether it was the reproductive state or the strain. So this is one thing, that listening to many imperfect neurons is more practical than finding the one expert. This is one general message that I'd like to make. And also, as I showed you before in the correlation analysis, a given unit can participate in multiple classifications or networks. So these are supposed to be the A or B neurons. This is the downstream neurons. And for example, this is detection of one trait, maybe a reproductive state. This is detection of another trait. And the same neurons may participate in different types of classifications depending on what the downstream neurons would want to listen to. Okay, so this is the general one of the conclusions. Now, you may say, and the reviewers have said, maybe there are single units that can do a perfect job, and you just didn't record from them. And my answer to this is that it's possible. Of course, we do not know. We do not sample all the neurons in the accessory olfactory bulb, but in reality, the task is actually much more difficult than we modeled here, because we just looked at a limited number of variability, of sources of variability, dilution and the strain and the state. But what about other things, like the age of the animal and how long the dilution was outside in the sun to evaporate and maybe oxidized and so on. So there are many more factors of variability which will make the problem more difficult and more unlikely that there is one expert neuron in the data set. And As I said, generally many non-experts, I think, are better than a small number of experts. And one of the analysis that we did to show that is to begin classifying with all neurons as we did before, but now at each stage, not to remove the least informative neurons, but actually take the one that gives us the highest weight, the one that's supposedly the most influential, and see how quickly the performance drops down. So this is similar to the curves that you saw before, but now we look at the number of neurons that were removed. We did this 10 different times, but it's essentially just to account for the randomness in the training procedure. And you can see that about between 20 to 30 neurons, you can still remove them. Okay, you can still remove the between 20 and 30 best units and still be better than the one best neuron that you have. So this strengthens my belief that it's best that even if you have a couple of neurons that are not optimal each in themselves, listening to several of them can help you a lot in making a good decision. Now, another thing that may be said, rightly so, that surely the brain is not a perceptron. The brain can do much better and is more complicated than a perceptron, and of course we agree. And one thing that we did here was also look at a more complicated classifier, a support vector machine, which is not a linear classifier. And indeed, for the same classifications that I showed you before, the support vector machine does do better. If you have many neurons, it can improve and also almost or reach 100% of the specific classification. So we're not saying that the perceptron is the way that the information is extracted, but still the main conclusions that we made before about the difficulty of more quadratic hand, but that's all I remember about it. Adi.
Yes, I'm a little bit disappointed because the claim to fame of the Vino system was that each neuron each neuron is a Jennifer Aniston cell or a Simpson cell, at least in the periphery, and now your message is that it's doing more complicated computations like in cortex or something.
I'm not saying complicated, I'm saying it's more random actually, and nobody said about Jennifer Aniston and the AOB, not that I heard of, but well,
the ultra specificity of the
yes, this is at the level of the sensory neuron. So first of all, we're one level removed, and actually I would like to, I didn't show this picture, but there's something paradoxical maybe here, because we do think of the vomeronasal system as some innate system, right, hardwired system, and this would suggest that it's a very organized system, right? But when you look at the AOB, it's really a mess. So this figure, to go back a lot of slides, if you look, this one here, okay? These are the glomeruli, these are the mitral cells. The mitral cells in the AOB, they send their axons not just to one, as in the main olfactory bulb, but to several, and the numbers vary a lot. This doesn't seem like an organized system at all. Now, maybe this pattern of mess is reproduced in different animals. I don't think so. So there's something that's a bit inconsistent with something very innate, and I think that maybe one way to account for this is to require some training and to sample these different...
I'm again confused, because in the main bulb, it sends to the cortex information, and converges, and complex computations occur. But here this thing goes directly to the business end, right? The amygdala.
It goes to the amygdala, but things happen in the amygdala as well, and to the BNST, and to the it's not.
So what's the big difference between the main and the accessory?
I don't know I actually don't know. It's one of the big questions. I think I don't know. I can start speculating, but this will take me expensive time that I don't have, and also. We're trying to address this in one of the next things.
We don't even know whether this is the best stimulus. I mean, it could be that these neurons are tuned to Jennifer Aniston and you're giving them pictures of cars, right? I mean, it's not clear that you are in the right space. I mean, you are generally in the right space, but it's not clear how well.
It's not clear how well, but we have a lot of evidence suggesting that it is involved in social stimuli, in social recognition, and these are social stimuli, and we know that they activate the system. So maybe there are other stimuli. For example, the predator responses. The reason we know about predator now is because in the lab we happen to have predator urine in the fridge, and I just tested them, and they were very strong stimuli. And then it turned out that many receptors in the Vino are also responsive to those. So yeah, I don't know. Maybe there's something in some fridge that's even stronger.
How many neurons altogether you have in the mitral cell?
I don't know. I would say thousands, a few thousands, but I'd have to check that number. Okay, so The brain is not a perceptron. It's also not an SVM, support vector machine, though support vector machine is better. And as Chaim asked me before, I stressed again that we did something even more crude. We just described each response as one single number. And clearly that's not the case. And well, we think this is not the case because the responses have some temporal dynamics. And this is one thing that Anat is starting to look at now. So here you have, this just shows you that different neurons have different temporal profiles. So this is one neuron responding to male. This is another one responding to predator urine. And you can see that they have different temporal profiles. Now, furthermore, a given neuron may actually have a different response, a different temporal response to different stimuli. Within one neuron, the response to female is more prolonged than the response to male. And likewise for this example. And it's even worse because for different concentrations, for a given stimulus and a given neuron, you may have different temporal response profiles. Okay, this is not as clean of an example, but you can see that it's more, the response is more rapid for a higher dilution, a lower concentration. So clearly there's a lot of information, or we think that there's a lot of information in the temporal features, and that's something that we haven't taken into account yet, and we have to take into account to see if this can improve declassification. Now here's another factor. Another simplification that we made is we gave just one stimulus. But when mice investigate each other, it's not just one activation of the pump, but it's something continuous, ongoing. This is from the classic experiments of Lowe and Katz. This is what brought me to the Katz lab in the first place. I don't know if it's also what brought Adi to the Katz lab. No, you were there before. But you were there before. But this is what attracted me to the lab. And in these recordings, we have responses of the mitral cell of one mouse as it investigates another mouse. And you can see that there's dynamics here and it takes a long time. Clearly, this is a series of many pump activations. And the next thing, or another thing that Anat has started looking at now, is what happens when we try to simulate several pump stimulations. So here we're looking at one stimulus in one neuron. This is just like before, one stimulation event. But here Anat stimulated 3 times, each separated by 8-second intervals, and here it's even more closely spaced 4-second intervals. And you see something that kind of makes sense. The response seems to add up as you have more stimulations. Here is another example, a nicer example, I think, where you have the predator urine, and then we stimulate 3 times at different intervals, and again you can see something that looks like some kind of a combination of these responses. But this is a response of the same neuron to a male stimulus. And this is something that I actually do not understand, and I asked Anat to check several times that this is a stable recording, and it seems to be. So we have here one stimulation to the male, but when Anat stimulates the nerve several times, actually the second stimulation seems to truncate the response. And this is true for both the higher interval and for the quicker interval, which is something I cannot explain, but it says something very important. It says that while these stimuli may elicit similar responses upon one stimulation, if you look at a sequence of stimulations, it's going to be completely different. And you could think that maybe that's a mechanism for the mouse to separate two things that are similar. This is very speculative, but this is just one example that's very interesting. And now I'm going to have to be even super fast because I see the time now. And I want to tell you something about learning and plasticity in the vomeronasal system, specifically in the AOB. And does anybody here not know about the Bruce effect? Yes, so let me quickly describe the Bruce effect. The Bruce effect, this is Michal Frankel, she's studying the Bruce effect, and this is the Bruce effect. And here's the effect. You have a female mouse, she mates with a male, she has a litter, the color of the male. And then she's exposed to a strange male, shown here in black, and in a very large percentage of high probability, her pregnancy will terminate. It will be blocked. Now, the interesting thing about this effect is that exposure to the familiar male does not lead to the pregnancy block, okay? So something happened during mating and the females learned to recognize, or the female's nervous system learned to recognize the stud male and prevent him from causing the pregnancy failure. And there's nothing special about the brown versus the black. I could have just reversed the colors or the identities. She could also be impregnated by the black male and then be immune, if you will, to his pheromones. Now, what's the current model? And this is a model that's been in the literature for a long time based on several studies. How does the Bruce effect work? So there are 2 elements. One is the block. And the idea with the block is that pheromones, stimuli activate the Vino, the AOB. The business end that Adi mentioned in the amygdala and the hypothalamus. There's activation of dopamine neurons that actually release dopamine in the pituitary. They suppress prolactin release, which is required for the pregnancy in the first days. And if you have the sequence of events, then there's a block of prolactin and pregnancy failure. This is the model for what the strange male does to the female mouse. It's even more interesting what happens with learning, so with mating. And the idea is that during mating, the mechanical stimulation of the cervix of the female causes activation of locus coeruleus, or noradrenergic cells in the locus coeruleus in the brainstem. This causes release of noradrenaline in the AOB, and the simultaneous presence of noradrenaline and the activation of the mitral cells due to the odors of that stud male, the mating male, cause some suppression. I'm going to describe this quickly. They cause a specific suppression of the odors of the stud male. Okay, so the idea is that after mating, the female will not smell the stud male through her AOB. There will be a suppression. It's called the negative template hypothesis. So his odors will no longer be able to trigger this sequence of events. What?
How long would it stay?
To what?
The suppression.
So the memory, you mean? The memory lasts for about 2 months from the best studies that characterize this. So it's called imprinting because it's a very long lasting. It's not a lifetime, but it's long lasting. And I'm hurrying up here because I want to show you some of this data. So these are the experiments that Michal planned and did. There's a very clear prediction that after mating with a certain male, then responses to this male will no longer be as strong as they were before. And what Michal did here, Amit, you have to stop me. What Michal did here was to take female mice, have them mate with males of various strains, and then check the responses to these different strains. And she did this with bulb C and C57 mice. And here it's important to say that the behavior is well established, Ifat, in the sense that we know that bulb C mice can cause pregnancy failure after female mated with C57, also vice versa. So we know that this is a good model to study this specific phenomenon. And here I'm showing you examples of recordings that Michal made. So this is a mated female, and you can see here the stimuli. The legend is a bit complicated, so I'll point to you the important stimuli. This bar here corresponds to the male, to the stimuli, to the urine from the male that this female mated with. So she mated with a C57 individual. We have 2 C57s, but this is one of them. And indeed, the bars show you how strong the response is. It's a summary of the PSDH. This neuron in this female responded to one of the female stimuli. Okay, nothing that contradicts the theory. It's as expected. But, yeah, sorry, these are all urinary stimuli. Now we're looking at another female, or maybe it's the same female, but here you see a broad response range. So in this neuron, you see a response to many of the different stimuli, including the predators and the females, and also to the mating male, which is not so much consistent with the idea of the pregnancy block. And here you see another example of a mated female, and again you see a very strong response to the mated male strain. Okay, you see it right here. The bars to the bulb C with which the female mated with are strong. I will skip this slide and tell you that what we think so far is that the basic model of the Bruce effect, and this is a very dominant model that's been around for many years. It's not such a clear-cut shutdown that we see. So something probably more complicated is going on here. There's not a complete suppression to orders from that specific strain. It's not entirely surprising given that many neurons respond to multiple stimuli. It's not surprising because given neurons respond to several stimuli. So if they would completely be suppressed, it would destroy not just the representation of the mated male, but also representation of other stimuli. So again, unfortunately, we do not have the answers yet to what is going on here. We probably know better what is not going on, and it's not a specific suppression of the male stimuli. These are very preliminary results. This is why I don't want my talk on the air, because we have to do some more analysis of this and to make sure and to see how the distributions change as a function of mating. Now,
reduction in the response to a familiar odor, but it's at the output
Yeah.
The F11 would be
This could be another mechanism, but his does not depend on mating, whereas the phenomenon of the Bruce effect is supposed to be. And there are many studies showing that things do happen at the AOB following mating. So EM studies showing them neurotransmitters levels change. Something is probably happening in the AOB. We don't know what it is. I have one more part. I don't feel I will stop now. I will just tell you what it is. We're trying to understand also whether we can. This is Karen who led this work. I will say this. The Bruce effect suggests that the vomeronasal system is a hardwired system, right? There's a hardwiring that leads to prolactin suppression. This is also the idea that Adi mentioned. We're trying to see if this is the case by doing one type of experiment. We're trying to see if we can train an animal to associate AOB activity with specific behavioral responses. And we're using optogenetics to do this, and we're comparing the male factory bulb and the accessory factory bulb. So far, with these complicated experiments, we were able, Karen and people that she was working with, were able to show that we can train the mouse to do this with the male factory system, and the fibers are positioned in place to do this now in the accessory factory system, and this is something that we'll be hopefully having answers within a few weeks or months. So just to summarize now my talk, I tried to give you the foundation of what we're doing in the lab, the general framework. I showed you the earliest, the more mature study in the lab about the encoding information by multiple neurons. I touched a bit on the issue of temporal coding. This is all Anat's work. Michal's work on the plasticity in the context of the Bruce effect. And this is the behavior that I did not have time to talk about, but this was led by Karen, and many students contributed, Rachel and Asmahan and Noah, who is a master student who is now becoming central. I did not have time to talk about the work of Xiao Peng and Oksana and Rohini, but these are also stories that we also think are very interesting. So that's the end of my talk, and thank you very much. Yeah, but it's still a problem of classification, except now you're classifying estrus versus, or estrus female versus anything else, right? It's always one option or another. And
it's more a problem of protection. For example, when you're straight, you just artificially gave them two different strains.
Of course, there are many strains. It's not a binary decision. This is a binary decision, and it's more complex than a binary decision, of course. The classification.
Would this improve anything?
I'm not sure. This seems to me, I don't know. I don't have a good answer off the top of my head, but I think that it is valid to think of this as a detection problem. I see. Let's talk about the female mouse now. Okay, there was another male. She has, not she, but it has to be decided whether this is a bulb female or a C57 male in order to, it doesn't work like this, but in order to terminate the pregnancy or not. So I think it can be framed as a term, as a form of a classification problem. But I'm not saying it's not a binary decision. It's not a binary decision. It's more complicated than that. And I'm not sure that this is actually how the system works. Yes. So. Yeah.
One more thing.
Yeah.
Sort of related to this. In your toy problem that you presented in the beginning with concentration.
Yeah.
That no threshold will determine. In the visual system, I think these things are solved with population normalization. Thought to be solved. Have you seen any sort of evidence in the formative
So I'm not sure what you mean, but for example, you can talk about a relational code, right? So you can normalize for the dilution, for example, by normalizing by some other component. And of course, if you have some metabolic pathway, maybe what's important is the ratio between two metabolites and not their absolute levels. So this could solve some of these problems that I posed and make them not really relevant problems. I don't know if it really happens. We do know that they have, for example, log linear responses that give you the conditions to maybe compensate for different concentrations. I don't know if that wasn't a good explanation, but the responses seem to be consistent with the ability to do a concentration normalization. Okay, but what really happens, we do not know. So it's a good solution, one that makes sense to me, and I think that probably that's how the system works. Maybe that's what mitral cells are doing, comparing levels of different compounds, or maybe downstream neurons are doing it. But this seems to me a good way to solve some of these things, and maybe those neurons that maybe our classifier actually even implements something like this, comparing different responses of different neurons.
One more question, anybody? I will have the last question. So I was wondering, you use the urine, which has many components, and also at the beginning you talked about one component against another one.
Yeah.
So, and especially for the last section, we talked about the hard experiment. Why don't you use one component that drives behavior, and then you are more able to see maybe what's the effect?
Because for many of these cases, we don't have one compound that drives behavior in such a way. There are a small number of identified compounds. Some of them have known effects, it's true, but not for all of the things that we study. We don't know what is the unique BALB3 or C57 stimulus. And the other thing is that I think this is the more real phenomena. So we have a problem here. We have to deal with something complicated. We're blind to what these stimuli are, but this is one of the challenges that we have to face. So yeah, it's a different way to tackle the problem. We look at natural stimuli in their complex.